Welcome back to the deep dive. Today, we are undertaking a pretty intense exploration into one of the brain's most critical and, well, often most confusing subcortical areas, the basal ganglia.
It's a phenomenal system. People call it the brain's main movement controller, but
Okay.
it's really less about starting a movement and more about, say, selecting which movements to allow and which to suppress.
So our mission today is to take a stack of anatomical sources,
Mhm.
specifically that core chapter on this, and really build you a mental map of how all these pieces fit.
Exactly. We're turning those complex, sort of intertwined nuclear masses into a clear, functional blueprint you can actually visualize.
Okay, so clarity over minutia. When we say basal ganglia, that term's been a bit broad historically, right? It used
Yeah.
to include things like the amygdala. But what's the functional club we're focusing on right now?
Right. So the really relevant group focuses on three main components that work together in a functional loop. First, the core is the corpus striatum. That handles the major inputs and outputs,
The in and out.
the in and outs. Then you have two crucial modulators, the subthalamic nucleus and the substantia nigra.
And the substantia nigra sits a bit lower down, doesn't it, in the midbrain?
That's the one. And if those three pieces aren't talking to each other correctly, well, movement fails.
And what does that failure look like? I mean, why is this such a critical area of study?
Because the consequences are they're immediately recognizable. This system is what smooths out our posture, our actions. When it malfunctions, you get the extremes.
Right.
On one hand, you get that poverty of movement, the stiffness and rigidity of Parkinson's. On the other, you get these dramatic, uncontrolled, involuntary motions, the dyskinesias.
So his whole job is to maintain that
A
balance.
perfect balance between starting and stopping. That's the defining role.
Okay, let's unpack that central anatomical structure then, the corpus striatum. When anatomists look at it, they see three distinct masses. What are those classic players?
So we're looking at the caudate nucleus, the putamen, and the globus pallidus.
Now, I know the terminology here can get tricky, and that's a huge source of confusion for students. The names don't always match the function, especially because of how close they are.
That's a key distinction you have to get your head around. For years, the putamen and globus pallidus were just grouped together as the lentiform complex.
Just because they sit side by side.
Exactly, just because of proximity. But we now know that chemically, functionally, the putamen is much more similar to the caudate nucleus.
So the caudate and the putamen are the real functional buddies here. What do we call them together now?
They're jointly called the striatum, or sometimes the neostriatum, and this is probably the most critical functional point to grasp. The striatum, that's the caudate, and the putamen, is the main receiving station.
The input structure.
It's the input structure taking signals from all over the cerebral cortex. The globus pallidus, on the other hand, is functionally separate. It's primarily the output structure.
Let's lock in on that input side first, the striatum, because visualizing these structures is really half the battle. How do we picture the caudate nucleus snaking its way through the brain?
You should think of the caudate nucleus as this large, dramatically curved mass, like a giant tadpole, or maybe a big parenthesis.
Okay.
Its fat, rounded head sits right up at the front, and it forms the floor of the lateral ventricle in the frontal lobe.
And it doesn't just stop there. It curves all the way around.
All the way. The head tapers back into a slender body, which then curves sharply down and forward, forming this long, thin tail. That tail follows the exact path of the lateral ventricle as it curves down into the temporal lobe.
So if you can trace the shape of the lateral ventricle,
You've essentially traced the caudate. It's an enormous C-shaped input structure.
and where's its partner, the putamen, in relation to all that?
So if the caudate is hugging the ventricles on the inside, the putamen is sitting more laterally, kind of pressed up against the side of the brain. The only thing separating these two functional partners is a massive sheet of fibers.
The anterior limb of the internal capsule.
That's it. This huge communication highway literally splits the functional input structure into two parts.
That's a great visual. Now, you mentioned something called the ventral striatum. Why is that smaller lower region so important for behavior?
The ventral striatum is continuous with the head of the caudate, and its most famous part is the nucleus accumbens. And when you hear about motivation, pleasure, reward, addiction,
That's where the action is.
that's where the action is. The nucleus accumbens is absolutely central to the mesolimbic dopamine pathway. It really shows how the basal ganglia doesn't just manage physical movement. It manages motivated movement, that drive to seek a reward.
Okay, so this is where we move into the control room, the output centers. Tell us about the globus pallidus, or the pallidum.
The pallidum sits right medial to the putamen, basically sandwiched between it and the internal capsule. And it's anatomically split into two segments, the lateral segment and the medial segment.
And those two segments do dramatically different things, right?
Crucially different. The medial segment is the true output stage. It's the final common pathway sending inhibitory signals out to the thalamus. The lateral segment, though, is mainly a relay station within the circuit, feeding information to the subthalamic nucleus.
Let's focus on that output stream then, the axons leaving the medial pallidus. How do those inhibitory signals finally get out of the basal ganglia to reach the thalamus?
Well, the output is actually quite complex because the fibers have to find a way either around or through that massive internal capsule. They exit the pallidus in two main streams.
Okay.
One stream loops around the capsule, that's the ansa lenticularis, and the other penetrates through it, the fasciculus lenticularis. Both of these bundles then quickly converge just below the thalamus in the subthalamic region.
And what happens there?
They make this really tight hairpin turn to go up into the thalamus as a single tract. This is the final signal that dictates whether or not a movement is going to be suppressed.
And that convergence point is where we find our next key player, the subthalamic nucleus, or STN. What's its chemical signature? Why is it so powerful?
The STN is physically small, it's biconvex, but what really sets it apart chemically is that its neurons are glutamatergic.
Meaning they're highly excitatory.
Highly excitatory. In a system that's almost entirely dominated by inhibitory GABA, the STN is this powerful, centralized accelerator. It fires excitatory signals back to the output structures, both the medial pallidus and the substantia nigra.
So we have the inputs, the striatum, the main output, the medial pallidus, and the central accelerator, the STN. This brings us to the core functional engine, the connectivity. We have these two competing loops, the direct and the indirect pathways.
Yeah, thinking of it like a car is a perfect analogy. Both pathways start in the striatum, and the cells there, these medium spiny neurons, are inhibitory. They use GABA.
Okay, let's start with the direct pathway, the accelerator. How does that one work?
The direct pathway is the simple route. It goes striatum straight to the medial pallidus. Now, because the striatum is inhibitory, it inhibits the medial pallidus.
And the medial pallidus is also inhibitory toward the thallus.
Exactly. So if you inhibit an inhibitor,
You get disinhibition. You're releasing the brake on the thalamus.
you're releasing the brake. The net effect is to promote the cortical motor loop. It's the accelerator, it supports the movement you want to make, and it uses D1 dopamine receptors.
Now for the brake, the indirect pathway. This one has a few more stops.
The indirect pathway is the more complex regulatory route. It's designed to suppress all the movements you don't want to make. It goes striatum to the lateral pallidus, then to the subthalamic nucleus, and finally to the medial pallidus.
Okay, so the striatum inhibits the lateral pallidus.
Correct. But if you inhibit the lateral pallidus, you release its inhibitory hold on the STN. So our central accelerator, the STN, becomes excited.
And since the STN is glutamatergic.
It fires a huge excitatory signal to the medial pallidus. This massively increases the inhibition coming from the medial pallidus to the thalamus. The net effect is a powerful brake on movement. And this pathway uses D2 dopamine receptors.
That is brilliant. So the accelerator takes a short route, and the brake takes a longer, more complex route to ensure movement is suppressed effectively. Now, where does the critical piece, dopamine, fit into this delicate equation?
Dopamine is the genius element here. It's supplied by the substantia nigra pars compacta. And it doesn't just apply or remove the brake, it does both at the same time to ensure you get seamless movement selection.
How does it do both?
Dopamine excites the D1 receptors of the direct pathway, so it's pushing down on the accelerator. And simultaneously, it inhibits the D2 receptors of the indirect pathway, which means it's lifting up the brake.
That dual action is the key to fluidity. Dopamine makes sure that when we want to move, we have maximum promotion of that movement and minimal suppression of anything else.
Exactly. It's the modulator that transforms a potential movement into a smooth, intentional action. And if you lose that perfect modulation, well, everything just unravels.
Which brings us right to the clinical side. Let's trace that unraveling in the most famous example, Parkinson's disease.
Parkinson's is, at its core, the catastrophic loss of those dopaminergic neurons in substantia nigra. When the dopamine is gone, the entire system just flips into a state of excessive suppression.
Walk us through that functional collapse.
So with no dopamine, the indirect pathway is no longer inhibited, so the brake is constantly jammed on, and the direct pathway is no longer excited, so the accelerator is disabled.
Gotcha.
This leads to an excessive activation of the STN, which then causes massive overactivity of the basal ganglia's outputting. That huge inhibitory signal just shuts down the motor thalamus and, by extension, the motor cortex.
So the symptoms of rigidity, tremor, that poverty of movement akinesia, it's actually caused by a basal ganglia that is wildly overactive in its ability to inhibit. It's too restrictive.
Precisely. It's a hyper-inhibitory state. Now let's consider the exact opposite scenario. Too much movement, or dyskinesias.
Right, like the violent, uncontrolled movements of hemiplegismus. Where does the lesion have to be to cause something that explosive?
Hemiplegismus is the classic result of a stroke or a lesion that destroys the subthalamic nucleus, the STN.
Our main excitatory pedal.
Exactly. If you lose the STN, that excitatory drive to the medial pallidus is just gone. And that causes the basal ganglia's output to become profoundly underactive.
So if the output is underactive, the brake is basically disabled.
The brake is off. You get massive disinhibition of the thalamus and cortex, which leads to these violent, flinging, uncontrolled movements on the opposite side of the body. In fact, a lot of other dyskinesias, like the ones you can get from high-dose L-dopa treatment in Parkinson's, are functionally linked to underactivity of that same indirect pathway.
It's incredible how this detailed understanding of the loops has just revolutionized surgical therapy. Tell us a bit about deep brain stimulation, or DBS.
DBS is really the ultimate testament to this anatomical model. We can now precisely modulate these structures. For Parkinson's patients, where the system is hypoinhibitory, surgeons will often place stimulating electrodes right in the subthalamic nucleus.
And what did that do?
By delivering high-frequency stimulation, they effectively inhibit that overactive STN. It cuts the excessive excitatory drive to the output, and that alleviates the rigidity and the akinesia.
And what about for patients with severe dyskinesias, where the system is underinhibitory?
In those cases, the target is often the main output structure itself, the medial segment of the globus pallidus. Stimulating or even ablating that area helps to smooth out the unwanted movements. It just proves that we can restore balance by intervening at different points in the circuit.
So we've really built a comprehensive picture today. We have established the input structures, the striatum, which is the caudate and putamen, the critical output structure, the medial pallidus, and these two competing pathways.
The direct accelerator and the indirect brake.
All dynamically modulated by dopamine from the substantia nigra.
And what's so crucial to grasp is that the specific chemical complexity of this system, that interplay of GABA inhibition, glutamate excitation from the STN, and the dual D1-D2 action of dopamine, is what makes smooth function possible. It's a mechanism built for dynamic selection, not just static on-off control.
It's a remarkable example of function following form. But despite the success of DBS being rooted in this rate-based anatomical model, this idea that it's all about inhibition versus excitation, there's still a pretty huge mystery, isn't there?
Absolutely. Think about the pallidotomy paradox. If dyskinesias result from an underactive basal ganglia output, why does surgically lesioning or stimulating the output structure, the medial pallidus, actually help?
It seems completely counterintuitive.
It does. And it suggests that the basal ganglia isn't just about the rate of firing, it's about rhythm.
The rhythmic patterns?
Yes. The current cutting-edge research is all about the rhythmic oscillatory activity, particularly these beta frequencies, that become synchronized and pathologically increased in Parkinson's disease. We're moving beyond simple rate models to understand how abnormal neuronal synchronization itself is causing the symptoms.
So that's the frontier.
That's the frontier. What specific dynamic patterns of oscillation are we truly modulating with DBS, and how does that finally explain these paradoxes?
What a fantastic concept to leave you with. That we've perfected the mechanical map, but we're only now starting to learn the musical score. Thank you for joining us for this deep dive into the anatomical basis of movement control.